TB is the leading infectious disease killer at the moment, and the recent surveillance reports have showed that more people die from tuberculosis every year than die from HIV or die from any other infectious disease. So it's critically important that we tackle tuberculosis.
And whereas people know about HIV, they know about malaria, less people know about TB. It's in some ways it's a silent pathogen. It's there in the community. It causes huge amounts of health problems, huge amounts of morbidity, and yet we have really underinvested and neglected this disease as a global health problem.
If we are to contain the global burden of TB, we need new tools and we need new diagnostics and we need new treatments and we need a vaccine. In order to achieve this, researchers have a key role to play.
Tuberculosis has always been there. We've never been close to eradicating TB.
In Western Europe, we've seen something of a decline in tuberculosis over the years, but TB is still a problem in many parts of the world. There's been a particular increase in the last 10 to 20 years or so due to a number of factors. The emergence of the HIV epidemic. The HIV knocks out a patient's immune system and makes them much more susceptible to tuberculosis. But also we've seen increasing rates of drug resistance.
We also know that the social cohesion in a society is incredibly important to reducing the rates of tuberculosis. That's associated with poverty, and so very often in parts of the world where there is civil conflict, you will see rates of tuberculosis as other infections start to rise. That's extremely important in terms of where TB went to, or more importantly, why it's not gone away.
Tuberculosis is an infectious disease that is acquired from person to person. It's fundamental to the control of TB that we understand transmission, because if we can stop transmission, then we have a chance in ultimately eliminating TB, as is the case with the goal of the NTB strategy. It is therefore of interest to me that we use the best technology available, whether it is whole genome sequencing, geographical mapping of where cases are, tools to identify clusters of cases, as well as computer programs that predict where transmission is happening and improve statistical approaches to pinning down exactly where transmission is happening. Those are the key steps we require. Increasingly, we are recognizing that transmission does not necessarily happen in people's households, that a lot of transmission, especially in high burden settings, actually happens out there in indoor congregate community settings. And therefore, knowing exactly where these congregate settings are and putting in interventions to stop transmission in those settings is the key to containing TB.
Although we are definitely seeing an improvement in outcomes in terms of tuberculosis, we are also starting to see issues such as drug-resistant tuberculosis, and that is a direct consequence of treatment regimens that have been ineffective.
There's two aspects to why drug resistance emerges. The first is wholly about the patient and the relationship between the patient and the clinician. So it's the drugs that are given and whether the patient has been given drugs which are going to work and whether they're the right drugs, and then whether the patient is able to take those in the way that we require them to. So it might be that the drugs make them feel ill so they don't want to take them, or it might be that the patient is leading a chaotic lifestyle and so isn't able to take the drugs on a regular basis. So that's what my clinician colleagues focus on. They have to manage that process. What I'm interested in and what my team look at is the evolution of drug resistance, or how it emerges at a biological level for the bug itself. And for me, it's a question of almost compartments. So TB is tucked away within the host, within the patient. So it's tucked away. It's a difficult site to get drugs to. And if you're going to, drug resistance develops when you treat an organism with not enough of the compound. So not enough drug is getting to the bug. So if you're tucked away in a compartment within the patient where the drug isn't going to get to you, then you're already giving a suboptimal dose of the drug. So the compounds that we use have to get over those barriers. For many years we've used a smear, that's Mycobacterium tuberculosis sputum put onto a slide and then used to identify Mycobacterium tuberculosis. That's a robust, simple tool, and has served us well for over 100 years. But it doesn't serve us well in the days when we need to understand drug resistance in a quick and efficient way.
The importance of diagnostics in the current era is that we want to be able to make a diagnosis promptly. We've got better and better about that. The introduction of molecular diagnostics such as GeneXpert have undoubtedly improved the sensitivity and also the speed of diagnosis. What's most interesting about areas such as that is that you need both a very good test, which in general terms can be used close to the patient, so a point of care test, but also you need to re-educate your healthcare workforce and individuals who are being tested, the implications of the test and the value of it. One of the big questions about treating tuberculosis is that because the mycobacteria is both a slow-growing organism and also because it's relatively hardy to being killed by antimicrobials, it takes a long time to treat and effectively cure TB. So, for example, the standard therapy to deal with latent tuberculosis is 3 months of treatment continuously. The standard treatment for drug-sensitive active tuberculosis is 6 months of treatment. These are drugs that have potential toxicities. That in itself is a problem because, again, if someone is feeling more unwell on the therapy, then there's a good chance they may stop treatment.
Unfortunately, we haven't seen any improvements in the treatment of TB really for 30 or 40 years. And whilst those are very effective trials and the regimen works very well in trial settings, as we've seen in the history of the tuberculosis epidemic, the regimen is inadequate to control the disease. So we do need new treatments. We need better treatments. We need safer treatments, shorter treatments, treatments that are easier for patients to take. There's been a number of trials published in the last 2 years looking at 4-month treatments, the idea being that a shorter 4-month regimen would be easier to take for patients, and if it could be shown to be as good as the current standard of care, then that would greatly improve outcomes in practice and make it easier for tuberculosis programs to roll out treatment for a larger number of patients. Unfortunately, those 3 large trials showed that none of the 4-month regimens that were being evaluated were as good as the standard of care. So we're still looking for shorter treatments that can result in better outcomes for tuberculosis. A lot of the work in trials at the moment is looking at how we can best identify the most promising regimens to take forward to large, expensive trials. And so some of the work that we've done, some of the research that's being presented in the BMC Medicine journal series is looking at better ways of doing clinical trials to identify those regimens to take forward to large phase 3 trials.
At this particular time, immune correlates have emerged as an important issue because we have begun as a TB community to invest more in developing TB vaccines. So if you look pre-clinically, there's now quite an exciting pipeline of vaccine candidates who are waiting to be moved into clinical trials. But what we don't have at the moment is any real way of assessing which of those preclinical candidates are going to be the one that works and to go forward, because we don't have a correlate of immune protection. And so, of course, we can't invest in efficacy testing for every one of those candidate TB vaccines. And so we need tools, we need biomarkers and immune correlates to really select the most effective vaccines and to progress their clinical development as quickly as we can. And the reason why we need to do this is because if we do want to end TB by 2035, then we're going to need an effective vaccine by the year 2025. Typically, we say it takes 10 years to develop a TB vaccine. So if we don't start doing something about this today, we're not going to have that vaccine candidate by 2025.
The WHO has had, together with national TB programs, great success in tackling the problem of TB. Over the last cycle of international programs to contain TB, millions, tens of millions, over 40 million lives are estimated to have been saved. As a result of this success and building on it in relation to the United Nations Sustainable Development Goals, the WHO has developed a new strategy which is based on the vision that we want a world that is rid of TB. The strategy is based on 3 key pillars, and these pillars are important, including the need to have better integrated patient-centered care, the need to have strengthening of programs and systems to support the delivery of interventions, and better research and innovation. Research and innovation is one area that those of us in universities have a key role to play and should contribute to the discovery of new tools so that we can achieve the goals of the NTB strategy by 2035.